Today we're going to talk about sensation and perception, as well as deal with the main senses. One of these days I'm going to take this lecture and actually divide it out because there is a lot of information about basically, especially the visual system as well as the auditory system. And really, they really do deserve their own lectures in their own right. But right now, I do have them kind of grouped together. In the future, this lecture will disappear and I will divide them out to do a little bit more about each one of the senses, but just a warning. Of course, here is your joke. Hey, did you hear about the guy with synesthesia? Yeah, he mixes up his senses, but he has great taste in music. If you don't understand that joke now, hopefully you'll understand by the end of the lecture. When we look at this image in front of you, I want you to try to answer the question, how many people are in this image? You can pause this for a second and try to answer that. If you notice, we have a few extra bodies than heads. So some people might say there are 5 people because there's 5 heads. Maybe some people realize that there's several more bodies. Let's see, how many bodies we have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So just counted 10. I guess I should have probably gotten the highlighter and showed you. But this is a little bit of an optical illusion of sorts. And isn't that interesting how our bodies are able to perceive, potentially very, very different things? This comes from a book called The Playful Eye. One of a really ginormous version of this was sitting in my office, and then people came and cleaned out my office. But I wanted to get a digital version of this so I can actually show it. Sensation versus perception is what we're going to be discussing here. While they are related, they are actually very, very different things. When you look over at this image here, you can pause this for a second and look at, what do you actually see? Well, if you're American and been around for the past few years, you'll probably see something like maybe Batman near a fire, or from a different perspective, you can see the Joker and his eyes. So the Joker's in white, Batman's in black. There are a lot of little visual things like this. Once you've seen usually you'll see one, then you'll see the other, and then when you're ever exposed to this again, you'll probably flip back and forth. There are other things like the old lady, young lady, but all of these are little games of, really perception. So what are these two different things? Sensation is, well, simply everything coming in, all the input from those external sources. In this case, maybe the light coming into your eyes. Perception is how you interpret all of this. Would somebody from another culture look at this image and would they see Batman and Joker? Well, they might or they might not, because the perception is that how we are interpreting the sensation, but it's influenced by a lot of factors, including experiences, culture, values, and all the other things on that slide. If you showed this to maybe a hunter-gatherer in Africa, I'm not quite sure what they would say. Maybe they'll see something. They'll see a guy with the fire, but would they see Joker? I really don't know. The sensation of all of this is, basically the same. They're going to see something similar. But are they going to see, specifically Batman and Joker? well, they're probably not going to perceive it that way. And we're in an interesting situation where sometimes we do have to be aware that other people's perceptions could very well be different. I could see something and, well, maybe not everybody will see the same thing. So let's give an example based on clothing. When I do this lecture, I frequently wear all black. So what is your how do you perceive somebody who's wearing all black? Why would maybe that's their favorite color? Maybe it means something very different. Maybe it means that person works in a job that requires all black. I was a theater technician for a good many years. It still holds a very strong place in my heart. And so I wore all black in high school. This is very different from, at the time there were also a whole bunch of other people that wore black, the goth crowd. I wasn't doing it for them. I was doing it because this is how my group, the theater techs, how we worked. We had to. It's really a bad idea to wear very bright colors when you're, say, moving set pieces, during a blackout. It doesn't work out very well. If you ever go to a hospital, maybe different colors will show up. Different scrubs might represent different departments. So it could also just be that. But going back to this whole thing about perception, so I'm currently recording this in spring 2021. A few months ago, there was a situation where President Trump got a little bit freaked out because a bunch of people dressed all in black boarded an airplane. They also had apparently tattoos or something like that. And he perceived them all as terrorists, when realistically they could have just as easily been theater technicians. In fact, I found out about this whole situation based on something on Facebook, where they joked and said, okay, which one of you guys, freaked this poor man out? He automatically assumed perception, he perceived these people as being dangerous, when me and a whole bunch of other stagehands just said, okay, so it sounds like a bunch of theater techs just work on a show, which we can fill up a plane, trust me. I want you to read this really, really fast. So if you read it really, really fast, read it out loud, you might not have you might have noticed something, or you might not have noticed something. Most of the time when people read this super, super fast out loud, they say, I love Paris in the springtime. It actually says, I love Paris in the springtime. Part of this is because, yes, we sensed all of these words, but it doesn't make a lot of sense to have the repeated twice. So all this stuff came in, but our brain only perceived and really noticed one of those thes. It's something to think about. So always be careful of what could be coming in, especially if you're doing something really fast. There are a few phrases that are also very, very helpful to know when we're discussing sensation and perception and such like that. The first is the absolute threshold. That's the absolute minimum needed to detect a stimulus. If you can't hear, something, let's say it's an audio thing, if you can't hear something under a certain level, that the level that you last heard it, that might be your absolute threshold. We do have some built-in absolute thresholds, in our body. Some things it's just we can't really deal with. But there is that absolute thing. There's also stuff like relative threshold and a little bit other stuff. But the most important here is the absolute threshold. There's also this thing called adjust noticeable difference, and that's the amount something has to be changed in order for you to be able to detect a difference, at least half the time. So let's say you're picking up some weights. Okay, let's say you're super strong, you're paying 100 pounds. You very well might notice it if I add 1 pound to you. You might notice it more if, say, it's a little bit lower. Say it's 10 pounds, you might notice a pound at that point. But, and I use that example because it's really easy to kind of visualize and understand in that way. But there's also do it in audio, which we're going to show there's going to be an audio, there's going to be a YouTube video in the next slide where you'll actually see that just noticeable difference a little bit better. But I did want to at least give one example. You also can see visually touch-based. Well, I mean, I guess lifting the weights would partially be touch-based. So that's the just noticeable difference. There's also this concept of Weber's law, which says the just noticeable difference is a constant proportion of the original value. Generally, people tend to think it's somewhere around 10%. So somebody who's carrying 100 pounds of whatever, like I said, they're probably not going to notice 1 pound. They're probably going to start noticing it if I give them 10 extra pounds, because it's somewhere around 10% usually. But these terms are terms that do come up on the PsycGRE, on the MCAT. They're not bad ones to know. On the next slide, we do have some more information about Weber's law and just noticeable difference. And, there was also some on the absolute threshold as well.
Well, here I am in my car, and I'm going to use my car stereo here to do a demonstration of absolute threshold, difference threshold, and Weber's law. Alright, there's a song playing, and I'm going to turn it up very slowly. Right now it's on 0, volume of 0. I'm going to turn it up very slowly, and the second you hear it, it will have crossed your absolute threshold. Now the volume's on 1. Do you hear it? 2. If you hear it now, it just crossed your absolute threshold. It's on 3. Now you probably hear it. It's at 4. It probably crossed your absolute threshold. 5, 6, 7. I hope you can hear it by now. It's probably crossed your absolute threshold. So the second that you can hear something, or see something, or feel something, the second that you detect it, the second it goes from nothing to something, it crosses your absolute threshold. Difference threshold is when you can detect a difference in a stimulus. So if I crank this up to like 20, it's pretty loud. If I change it 1, it's hard to tell a difference. Can you tell the difference between 20 and 10? I'm sure you can. You can probably tell the difference between 20 and 10. If you can tell a difference when I turn down the volume, it's crossed your difference threshold. Now, it's easier to tell a difference between something like 5 and 10, right? 5 to 10. You can tell the difference. But I'm going to turn it all the way up to 25 and see if you can tell the difference between 25 and 20. Or how about 30 and 25? Harder to tell the difference when a stimulus is really loud, or really bright in terms of light or really smelly in terms of smell. Well, here in terms of sound, it's harder to tell the difference between 20 or 30 and 35 than it is to tell the difference between like 10 and 15. Easy, right? Now 30 to 35. So that's Weber's law. Weber's law shows that it's easier to tell the difference between a low stimulus, and it's harder to detect a change in a really loud stimulus. There you go.
So for those who've seen my other lectures, I do like to have some activities here, and I've got a game called True or False. These are all vision related. There is an awful lot about vision, and this really should be its own lecture, which I'm probably going to say at least one other time during this lecture. Here's true or false. On a dark night, the human eye can detect a candle at 30 miles away. True or false? If you said true, you're correct. Actually, you might be able to go even further. Yes, there are people with poor vision, but even somebody who's nearsighted might be able to detect the light. It just might not be very, very clear. A baby's eyes are the same size they will be as an adult. True or false? If you said true, yeah, you're correct. They're basically the same size they're going to be as an adult. There is a little bit of wiggle room. So some people get a little bit of eye growth around the time they hit puberty, causing them to become slightly nearsighted. But because it is a slight growth of only a few microns, somebody can come in and fix their eyes through LASIK, just zapping off literally a few layers of cells, and their vision can be restored. All babies, when in the womb, have blue eyes. True or false? Because babies in the womb might not always have pigment, the answer is true. Blue eyes are caused by a lack of pigment. A lot of babies are still born with blue eyes because sometimes it takes the body a little bit of extra time to build the pigment so that they can darken up. I have dark eyes. I was born with blue eyes. My daughter was born with gray eyes. She actually has kept her gray eyes. Cultures perceive colors differently. True or false? While for the most part they all sense colors the same way, they might not necessarily perceive them the same way. For example, the Russians have two different words for blue. The Greeks, on the other hand, don't really have a blue. They call blue and green the same thing. It's actually kind of interesting. Also, and here's one that's English-centric, the color pink is specific to English. Most other cultures do not distinguish the color pink. True or false? The eye helps regulate your sleep and wake cycle. If you answer true, you're correct. Light coming into the system does, through a variety of different ways, actually trigger the perception of light, really triggers your circadian rhythm. True or false? Reading in dim light will hurt your vision. If you've said false, you're correct. There really isn't any evidence that reading in dim light will hurt your vision. Also, sitting too close to the TV probably won't do much either. It may cause you to have a little bit of eye strain, but that's not necessarily going to hurt your vision. So now it comes down to what is vision? Again, as I mentioned, I need to have this be its own section. So what is vision? What we have over here is a photo of our eye, or somebody's eye, maybe not your eye, where you see we've got basically the cornea, basically the iris and the pupil. Pupil's that big old black thing in the middle of it. The iris is the thing that's got all the different colors, like blue eyes, green eyes, whatever. Got your lens, and this is all basically your retina. Your retina, at the back part of the retina, we've got this area called the fovea, and here is where some of the photoreceptors hang out. Like right around here has a very high collection of a certain type of photoreceptor called a cone. We'll get more into cones later, but they help you see color. Here on these edges is going to have a different photoreceptor called rods. We'll get to them later. They're much better in low light conditions than bright light conditions. You may have also learned that if you when something comes into your eye, it actually projects upside down, and that's true. Comes in right here, for example, projects maybe there, and coming in on the other end there. Now, eventually this information does get converted and flipped back around in your brain, but it doesn't happen here. So anyway, so these photoreceptors go through a period of activation, and they pass that information on to the nervous system, and it goes through these areas like the optic nerve. By the way, this optic nerve produces a little bit of a blind spot right here, just so you know. Sometimes that comes up on the tests. Optic nerves go through, goes off to the brain. It has several places it can go to interpret, depending really on what it's looking for. There's something called LGN, which with multiple layers, and everything detects something different. Some detects motion, some detects color, stuff. But that's a whole nother. That's a lecture, literally in and of itself. So, but eventually, you go off, have this optic nerve, and you do figure out what's going on, and then you get to the point of awareness.
When you look at this painting, what do you see? A woman looking out a window? How about now? This famous painting by Salvador Dali is based on something called the Lincoln illusion. The effect shows how blurring pixelated images can make it easier to recognize faces. Optical illusions are fun to look at, but they can also tell us a lot about our sense of sight. So, how does the eye work exactly? And is it really true that seeing is believing? The human eye is pretty extraordinary. It has more than 2 million moving parts and can move faster than any other muscle at less than 1/100th of a second. But when it comes to your vision, the way you perceive the world is actually thanks to your brain. First, light enters the eye through the pupil. The muscles behind the iris squeeze and stretch the lens to focus the light onto the back of the eye. This is the retina. The retina is covered with sensitive photoreceptors called rods for dim light and cones for bright light. The focused light hits the retina at a small pit called the fovea, which has the highest density of cone receptors. We often compare our eyes to cameras, but they actually work quite differently. Our peripheral vision is very low resolution. We only see in full resolution at the small point of our fovea. But we barely notice it because our eyes are constantly refocusing on what we want to see. like a high-resolution spotlight. Pixels, or picture elements, are the tiny illuminated squares that make up a digital display. If 2 pixels are close to your eye, the retina sends 2 signals for the brain to interpret. Farther away, 2 pixels prompt 1 signal. That's why a TV screen can have a lower pixel density than a smartphone, but seen from farther away, it still appears seamless. When stimulated, the rods and cones send signals to the optic nerve and back to the brain. Then, all of that visual information is processed to create the picture you see of the world. Unlike a camera, your brain can actually fill in missing information. For example, your eyes automatically blink every 3 to 4 seconds. In fact, that means your eyes are technically closed for roughly 10% of your waking hours. Thankfully, your brain fills in those gaps, so you don't feel like the lights are flickering on and off all day long. There's a lot we still don't understand about the complexities of our visual system. As technologies continue to innovate, the line between illusion and reality could start to blur. And when it comes to how we see the world, there might be more than meets the eye.
As we mentioned, the photoreceptors, I already mentioned we have rods and cones, and I believe that was also mentioned in that little video, which I hope you guys enjoyed. But you might look at these 2 photos and realize that they're basically the same, just at different times of day. This top one is what we're going to see if we look at it at night. Mostly we're in this situation, you're going to have our rods are more likely to be activated because they're hopefully going to have been dark adapted. And here is what we're going to see during the daytime. So the rods, they're really more active in the dimmer light, and they are focused also a lot on our peripheral vision. They're not quite as clear as if we looked at something dead on, like when we saw the cones. See the cones. Cones are much more active in bright light. These are really what are being activated when we're looking at color. And that, yeah, you see in that top vision, yeah, there is a little bit of color, but though there's a little bit of blue, a little bit of orange around here. But those rods really can't see color. The cones are mostly focused in the fovea, and that's where the highest concentration is. But they're around here and there in other places throughout the back as well. But like I said, they do come in bright light. They are responsible for the super fine detail. Like, there's lots of things way in the back of this foot. Let's see. So a lot of things in the back you might not even have noticed. There's stuff right there. You can see a whole lot of really small details over there. Let's remove that stupid thing. But you see all sorts of things along, all the, I guess, people walking or whatever they're doing. You can see that on the bottom one. Our cones are allowing us to see that much more than our rods are. As we're discussing stuff like photoreceptors, well, let's look at this. I don't know why this I spelled color in a different way than everything else. Anyway, so chickens normally have 4 photoreceptors. The shrimp have 16. Can you imagine what it's like to be a shrimp? Dogs will tend to have basically 2. and humans typically have 3. So it's clear that the amount of photoreceptors actually vary by species. Now there are some humans that appear to have 4, and there are some humans that really only have 2 functioning ones, or potentially only have 1 functioning photoreceptor type. One of the theories of how we see color goes to, it relates to this thing called trichromacy theory. Basically, there are 3, tri means 3 color theory. And they have this S, M, and L cones. They're so named because they're sensitive to different types of wavelengths. So the smaller short cone, the S cone, would be more sensitive to things that have a shorter wavelength, like blue. Medium, M, be the medium wavelengths, like green. L, the longer ones, such as red. Now this seems to work kind of well with what we know about light theory. If you know much about light and how it works, unlike in art class where we have primary colors being red, yellow, and blue, light is a little bit different. In light, it's red, green, and blue as the primary colors. If you mix all these together in different amounts, you can get pretty much any color you want. So there is that. So there is another theory about how we see color, and there is support for both of these theories, by the way, is this opponent process theory of color, which says, yes, we have the 3 types of receptor, with a specific color receptor, but each one does one color and then that color's opposite, such as red can also sense green, blue can do yellow, and there's one that basically does black and white. So there is some support for this, especially when you look at things called color deficiency, visual color deficiency, where red and green are easily mistaken for each other. That's actually a very form of color deficiency. Color deficiency, by the way, is also called color blindness, but we are really trying to move away from that term because it really is a deficiency. They can see color, just sometimes some things not as clear as others. But red-green color deficiency, very, very common, especially in men. Blue and yellow is also another common deficiency, although it is my understanding that's a little bit more mixed as far as whether it's men or women. I think that's supposed to be closer to 50% is my understanding. And then we also have black and white, although black and white is its own form of color deficiency. Basically, they can pretty much only see that type of shades. But this is certainly one of those areas that people are very, very interested in. And because there are these different types of color deficiency, I do have something I'm going to show later about the different forms of color deficiency so you guys can understand. Okay, the first thing I'm going to discuss before I get to that color deficiency, which I believe is the next slide, is this concept of dark adaptation. This is a standard difference in vision. It's more unusual. It's not our usual mode, but it certainly is very, very common. In this case, the pupil is dilated, the rods become activated, and we start being able to see better in a dimmer condition. That's called, we're adapting to the dark, quite literally. For those of you who like doing amateur astronomy, you guys are probably going to be really familiar with that, because you try to get your eyes adapted to the dark, and so you can go observe and really appreciate what you're doing. For those of you who have friends who are amateur astronomers, I'm going to suggest to you right now that if they're trying to get themselves dark adapted, be very nice and try to avoid anything really, really bright around them. because while it can take 20 minutes to really adapt, it can just take a few seconds to lose. Trying to preserve your dark adaptation, I have a degree that's basically in what I call astronomy without the math. It's space studies. I did a lot of astronomy work. One of the tricks is to, if you're in a situation where you're trying to make yourself dark adapted, you're gonna have to start using basically red light because you don't lose your dark adaptation, as opposed to if you use like a bright flashlight when you're off observing. You can actually buy these little red flashlights, which are specific to help keep your dark adaptation. Also, if you're ever having LASIK done, red cellophane can actually help reduce your pain, because when you do get LASIK done, you get your eyes get very dilated, and they keep it that way. Basically, tricks your brain into thinking it's, well, permanently dark adapted, except for the fact you're not. You're often in bright light. So try to keep that in mind. But that is a temporary phenomenon, although with the LASIK stuff, it makes you feel like it's longer. So here is color vision deficiency.
How colorblind people see the world. Everyone's heard about colorblindness. A significant number of global celebrities such as Bill Clinton, Keanu Reeves, Mark Zuckerberg, and even Prince William are known to be colorblind. But do you know what colorblindness is? Colorblindness, also known as color vision deficiency, is not a disease, but a decreased ability to see color or differences in color. Approximately every 1 in 12 men and 1 in 200 women are affected by it. So, there's a high probability that someone you know, a neighbor or a co-student, is colorblind. There are 4 different types of colorblindness. Each of them provides deficiency in different color shades. For a person with normal vision, it is hard to imagine what it is like to be colorblind and how people like this see the world. Which color do they see? Do they see any at all? What does it mean not to be able to see red? And so on. That is why we at Brightside prepared a visual comparison of all colorblindness types and put them together for your better understanding. Let's have a look together. People with normal vision usually see the world around them in these colors. However, people with deuteranopia, the first type of colorblindness, see the same picture like this. Deuteranopia is the most common form of colorblindness. Around 4.63% of men suffer from it, and in many cases don't even realize it. It's clear from the photo that the colors have lost some of their brightness, especially with regard to green and red. And people with protanopia see these pencils like this. Protanopia is a less widespread form of colorblindness. Only around 1% of men experience it. All shades of green and red appear somewhat faded, and blue and yellow shades remain virtually unchanged. The third type of colorblindness is tritanopia, and they see colors like this. Though a very rare type of colorblindness, tritanopia affects both men and women to an equally small degree. Those who experience it see the world in greenish-pink tones. The last type is total color blindness. It is hard to imagine, but it exists. However, only a small percentage of people are affected by it. Only 0.00003% of the whole world's population. And now let's have a look at the straight comparison. How about a piece of fruit? Normal vision. Deuteranopia. Protanopia. Tritanopia. And together. Impressive, isn't it? And now, traffic lights. It is widely known that people with color vision deficiency can have difficulties while driving. By looking at these pictures, you will understand why. Here is normal vision. That's deuteranopia. This is protanopia. And that's tritanopia. And together again. Quite an eye-opener. A popular soda label, Normal Vision, Deuteranopia, Protanopia, Tritanopia. Together, my world will never be the same. What about a rainbow? Rainbow with normal vision, with Deuteranopia, with Protanopia, with Tritanopia. Together. So Richard of York gave battle in vain. Indeed. Street traffic. Look at the streetcar. Normal vision. Deuteranopia. Protanopia. Tritanopia. Together. And now the final example. Frida Kahlo was a Mexican painter in the first half of the 20th century. Was she ever thinking about how her paintings could be seen by colorblind people? Well, we don't know about Frida, but you are going to see it right now. Normal vision. Deuteranopia. Protanopia. Tritanopia. And together. Beautiful! Are you still with us? Then here are some historical facts for you. Colorblindness as a vision deficiency was discovered by English chemist John Dalton after he realized that he was colorblind himself. In 1798, he published the first scientific paper related to this subject, which was called Extraordinary Facts Relating to the Vision of Colors. Due to his immeasurable input into the research of colorblindness, the general condition has been named after him and known as Daltonism. Though, as Wikipedia states, in English, this term is now used only for deuteranopia.
So, if you actually want to learn more about John Dalton and hear about the paper, which apparently was a very interesting read, Stuff You Missed in History class in January 2021 actually did an entire episode dedicated to him. And I listened to it. It's actually kind of funny the way he described how he discovered he actually had a color deficiency. So, next up is something very, very different. Lord. The tetrachromats have something very, very different from the color deficient individuals. The tetrachromats actually have 4 cones in their eye, up from the usual 3. This is found mostly in women, actually almost exclusively in women. There are a few men that have it, but they're considered very, very anomalous, as in, I believe that's actually what they're called, anonymous tetrachromats. Usually in this case of women, they get it from their mothers, and usually they also have a male relative that usually has at least some slight deficiency. There's still a lot of research being done on tetrachromats. Now we enter the auditory system.
The sense of hearing is accomplished by a process known as auditory transduction. The ear converts sound waves in the air into electrical impulses, which can be interpreted by the brain. As sound enters the ear, it passes through the external auditory canal, where it meets the tympanic membrane. The tympanic membrane then vibrates in response to the sound. Sounds of a lower pitch, or frequency, produce a slower rate of vibration, and sounds of lower volume, or amplitude, produce a less dramatic vibration. Higher frequency sounds produce faster vibrations. The tympanic membrane is cone-shaped and articulates with a chain of 3 bones called the auditory ossicles. They consist of the malleus, the incus, and the stapes. The movements of the tympanic membrane vibrate the ossicles, passing on the information of frequency and amplitude. The 3 bones pivot together on an axis shown here in red. The pivotal axis is due to a series of ligaments which hold the bones in place within the middle ear cavity. The anterior malleal ligament and the posterior incutal ligament are of particular importance for the pivotal axis. 2 structures which normally obscure this view of the middle ear have been removed. They are the chordae tympani nerve and the tendon of the tensor tympani muscle. Through the ossicles, the vibrations of the tympanic membrane are transferred to the footplate of the stapes. The stapes moves with a piston-like action, which sends vibrations into a structure called the bony labyrinth. The labyrinth is filled with a fluid called perilymph. If it were a completely closed and inflexible system, the movement of the stapes would be unable to displace the perilymph, and therefore unable to send vibrations into the bony structure. Due to the flexibility of a membrane called the round window, the stapes movement can displace the perilymph, allowing vibrations to enter the labyrinth. The corridor leading to the round window is found within the spiral portion of the bony labyrinth known as the cochlea. Vibrations produced by the stapes are drawn into the spiral system and return to meet the round window. The portion of the spiral passage in which vibrations ascend to the apex of the cochlea is called the scala vestibuli. The descending portion of the passage is called the scala tympani. A third structure, called the cochlear duct, is situated between the scala vestibuli and the scala tympani. The cochlear duct is filled with a fluid called endolymph, and when viewed in cross-section, the membranes separating the 2 fluid-filled systems are visible. They are Reissner's membrane and the basilar membrane. The membranes are flexible and move in response to the vibrations traveling up the scala vestibuli. The movements of the membranes then send vibrations back down to the scala tympani. A specialized structure called the organ of Corti is situated on the basilar membrane. As the basilar membrane vibrates, the organ of Corti is stimulated, which sends nerve impulses to the brain via the cochlear nerve. The actual nerve impulses are generated by specialized cells within the organ of Corti called hair cells. The hair cells are closely covered by a structure called the tectorial membrane. As the basilar membrane vibrates, the tiny clusters of hairs are bent against the tectorial membrane, triggering the hair cells to fire. The entire basilar membrane does not vibrate simultaneously. Instead, specific areas along the basilar membrane move variably in response to different frequencies of sound. Lower frequencies vibrate the basilar membrane closer to the apex of the cochlea, whereas higher frequencies produce vibrations closer to the base. This arrangement is known as tonotopic organization. Together, this sequence of events is responsible for our acoustic perception of the world around us.
And luckily, that video basically takes us through what this slide is all about. Information comes in here, bounces through the canal, hits the membrane into the inner ear and the little bones there, and the middle ear rather, then goes off to the inner ear, and it eventually goes to the auditory nerve and goes to your brain. And that video also covered this, a little better detail as to where it goes from here, like I said, off to the brain. Much better than I could have done. Animation is a great thing. And we also, so now the auditory system actually also deserves its own lecture. And another sense is the sense of touch. And touch is kind of interesting. This is one that there are so many receptors that if you go off to like medical school or something, you're going to learn so much about these receptors. But in total, there are 6 different receptors that come into the that help you with the sense of touch. Those things include all sorts of things, such as they sense for position, vibration, fine or gross touch, pain, temperature. Excuse me. They feel a whole lot of things right here. So when things do go in, they eventually go through all the various receptors into the nerves. And as you see where this spinny thing here is, it does end up going to the primary somatosensory cortex. You may have heard me talk in other lectures about in the parietal lobe, every little body part is represented by a different part of that parietal lobe. This and I said there was basically a little human figure that you could use, that homunculus, and that's what this is here. If you look carefully I can't stop this thing from spinning. You'll see where every little part is in there as to what it's controlling, touch or not. But it's kind of an interesting thing. So I'm gonna just let you guys think about this question and ask if you've ever lost your sense of smell and lost your sense of taste. Because I'd say most of us probably have. Smell and taste are what are called chemical senses, basically. And what's going on is at a point they do end up connected, but at this moment they're separated. When you're dealing with the sense of smell, basically we've got the compounds that have the smell, the odorous compound. It's usually some type of fat-based thing because it does make much easier to sense. So it is usually lipid soluble of some form. Otherwise, I'm not entirely sure we'd be able to sense it. And so things come up from this. Don't ask me what in coffee is lipid soluble. I don't know. But it goes through, bounces through the inhaled air. Only about 10% or so of the compounds are actually going to get to this olfactory bulb here. And over here somewhere, they get absorbed by, they react to basically the receptor cell, which is where they're kind of absorbing. They dissolve into this mucosa, and it translates to the bulb, then to various other parts of the thing. It goes to the olfactory tract, etc., and eventually goes to different parts of the brain where it is processed. Now, taste, a lot of us have learned that taste has, well, that our tongue is 4, and this image actually is out of date. This is coming from the textbook, but I haven't updated anything yet. And well, mostly because I don't think the textbook really was is as up to date as I would like anyway. But in this, you may have seen this, saying one taste we can sense here, we can do others on different sides of the tongue, and I think it's supposed to be bitter that's in the back. Not only are there more than 4 tastes, there's actually at least 5. Some people actually say 6, but it's not that a specific part of the tongue actually specializes in this, that, or the other. The taste receptors, taste buds, are actually kind of all over the place, pretty evenly dispersed. But they do have, we do hear about, basically sweet, sour, sweet, sour, salty, and bitter. There's actually also umami. It's this like savory thing. I'm never ever sure how if I'm ever pronouncing that right, but I keep hearing it pronounced umami, and I always was wondering what the heck is that supposed to be. But apparently it detects glutamate, basically, and that is what it's actually looking for. Each one of these things reacts in a little bit different way. Like sour, we're actually detecting things like this. I believe it's not the high amount of like hydrogen bonds, which is how it reacts. Now, outside of the umami, there's also some people that actually say fat is also sensed. I'm not so sure. I'm on with fat as the 6th taste sense, because it really isn't a taste. They were saying it was maybe a texture, but I did like mentioning that because, well, I like giving as much information as I actually can. But in flavor, basically there are parts of the brain that also handles that flavor aspect, and they're processed not terribly far away from basically smell. So sometimes when you lose your sense of smell, your taste might also be affected, just because of how that they enhance each other. Really, I guess the smell enhances the taste. There are also types of things called multimodal perceptions, such as the senses that can actually affect each other and help each other out. Like taste and smell can help each other out. But there's also this extreme version called synesthesia, which is basically they're a little bit, overlapping a little bit too much, and it's actually a very interesting experience to hear about. And let's see about that. And now, synesthesia.
Imagine a world in which you see numbers and letters as colored, even though they're printed in black, in which music or voices trigger a swirl of moving colored shapes, in which words and names fill your mouth with unusual flavors. Jail tastes like cold, hard bacon, while Derek tastes like earwax. Welcome to synesthesia, the neurological phenomenon that couples two or more senses in 4% of the population. A synesthete might not only hear my voice, but also see it, taste it, or feel it as a physical touch. Sharing the same root with anesthesia, meaning no sensation, synesthesia means joint sensation. Having one type, such as colored hearing, gives you a 50% chance of having a second, third, or fourth type. 1 in 90 among us experience graphemes, the written elements of language like letters, numerals, and punctuation marks, as saturated with color. Some even have gender or personality. For Gail, 3 is athletic and sporty. 9 is a vain, elitist girl. By contrast, the sound units of language, or phonemes, trigger synesthetic tastes. For James, college tastes like sausage, as does message and similar words with the "ige" ending. Synesthesia is a trait, like having blue eyes, rather than a disorder, because there's nothing wrong. In fact, all the extra hooks endow synesthetes with superior memories. For example, a girl runs into someone she met long ago. Let's see, she had a green name. Deezer Green, Deborah, Darby, Dorothy, Denise. Yes, her name is Denise. Once established in childhood, pairings remain fixed for life. Synesthetes inherit a biological propensity for hyperconnecting brain neurons, but then must be exposed to cultural artifacts such as calendars, food names, and alphabets. The amazing thing is that a single nucleotide change in the sequence of one's DNA alters perception. In this way, synesthesia provides a path to understanding subjective differences, how two people can see the same thing differently. Take Sean, who prefers blue-tasting foods such as milk, oranges, and spinach. The gene heightens normally occurring connections between the taste area in his frontal lobe and the color area further back. But suppose in someone else that the gene acted in non-sensory areas. You'd then have the ability to link seemingly unrelated things, which is the definition of metaphor, seeing the similar in the dissimilar. Not surprisingly, synesthesia is more common in artists who excel at making metaphors, like novelist Vladimir Nabokov, painter David Hockney, and composers Billy Joel and Lady Gaga. But why do the rest of us non-synesthetes understand metaphors like sharp cheese or sweet person? It so happens that sight, sound, and movement already map to one another so closely that even bad ventriloquists convince us that the dummy is talking. Movies likewise convince us that the sound is coming from the actors' mouths rather than surrounding speakers. So, inwardly, we're all synesthetes, outwardly unaware of the perceptual couplings happening all the time. Crosstalk in the brain is the rule, not the exception. And that sounds like a sweet deal to me.
And that is synesthesia. And here are your discussion questions. So, what sense might you most hate to lose? Which one would you be most willing to lose? Why? How would our ancient ancestors answer that previous question? And have society and technology made certain senses less essential to survive? Which sense is most connected to your sense of identity or self? Those are more the philosophical ones, I suppose. But how does brain code for body areas that might be more sensitive than others? And does anybody know what would happen if the input from a limb to the primary somatosensory cortex, which detects touch, was destroyed through something like amputation? Let you guys think of some of those questions. And if you have any questions for me, you can feel free to drop an email, drop a comment, whatever you like to do. And I will see you guys later.